I think it's always true in science that when you're surprised, it's almost the best opportunity to make a true discovery.
The vast majority of tumors that humans have are based on the misregulation of stem cells. Colon cancer, pancreatic cancer, leukemias. One of the unsolved problems in stem cell biology is understanding how these cells are regulated by their environment.
Most stem cells have managers or micromanagers. Planarian stem cells have found a way to be independent. Most of the stem cells we know about from other organisms reside in a location called a niche, a physical place in tissue where other cells surround the stem cells and support them and give them instructions.
We wanted to know if there was a specific neighborhood, a specific microenvironment that exists in planarians that makes them so special.
Planarians have the amazing ability to regenerate after any injury. They can replace any missing tissue of the body and even an entire body from a very small fragment.
We thought we would just find one microenvironment or one neighborhood where stem cells existed that would explain all of this, but it's actually much more complicated.
We didn't know where to look because there are so many stem cells in the planarians that they are near almost everything.
So we decided to use this brand new technique where we could map spatially all sorts of different factors in planarians and figure out their relative position to stem cells.
We were surprised to find that what was next to stem cells most often was not anything we had characterized before, but instead a new uncharacterized cell type that we got to name hecatonoblasts. We named the cells after a monster from Greek mythology, the Hecatoncheires, because like the monster, the cells are very large and they have lots of long projections, or in the monster's case, arms. We thought the hecatonoblasts were going to be controlling the stem cells. We were surprised to find that hecatonoblasts are not required to direct regeneration or even to do their basic functions like dividing. Instead, we found that more distant cells, such as the intestine, are controlling the activity of the stem cells, meaning that planarian stem cells are not controlled by their neighbors, but rather over longer distances in ways that are foreign to what we've seen before.
When the population needs to do a big thing, like rebuild a head after the worm has lost its head, it has to know what's going on across the whole organism. Human patients are not very good at maintaining tissues throughout their lifespan. So the more we understand how local microenvironments and global signaling events can be integrated to increase the potency and capacity of our stem cells, the better we'll be able to create paradigms to increase the regenerative capacity and hopefully regenerative medicine and therapeutics in humans.
The results were surprising and unexpected, and at some points completely backwards to what we thought we would find. This is what makes science so exciting, to really chase a hypothesis and try to answer it as correctly as possible. Sometimes we get to be surprised, and those are in fact the most fun moments.